So the hippocampus, a brain structure that's particularly relevant for today's meeting, is actually very small in the human brain, occupying about 1% of the size of the neocortex. And this sort of, and we know that it's actually critical for memory processing and cognitive function. And this suggests that this brain structure is bringing something to the table in terms of processing that the cortex cannot do. And so the overarching aim of this research program is to really try and identify these functions from a bottom-up perspective, with an emphasis on episodic processing, and then manipulate these functions to isolate what their role is in episodic processing. In addition to this sort of circuit level analysis will uncover new neuroscience, new phenomenology, provides a robust test bed for network level pharmacology, which is an emerging field and critical for drug development, as well as, last certainly but not least, providing the information required to develop novel theories and computational simulations of hippocampal function. We've had some experience of this sort of network level approach, some previous work in field CA3 of the hippocampus, where we manipulated local circuit operations within the subfield. And what we found was that field CA3, in response to a very brief input of 2-second pulse at 5 Hertz, was capable of generating this very prolonged reverberating activity. And this would go on for spiking for minutes after the initial stimulation. We then looked to see how this reverberation contributes to episodic processing. And this is using some novel behavioral tests developed by a former colleague in the lab, Dr. Brittany Cox, in combination with inhibitory dreads. We demonstrated that partially silencing this associational network completely impairs the ability of rodents to process temporal order, whilst leaving the semantic and spatial encoding intact. And so this was a really cool finding, linking a network function with the behavior in an episodic context. If we move on from this and extend this analysis to the entire network, this diagram needs no introduction to this audience. Obviously, the hippocampal circuit. Really, it's up here to remind me to mention that these inputs to this circuit arrive at specific frequencies or combinations thereof. And that's a theme that I'll come back to during this talk. But we know in terms of signal processing for episodic-like tasks that a number of processes are required. And these are listed here. We have signal transformation. So this is relating to the ability to filter or amplify an input. Cycling activity, which I referred to in the previous slide in terms of reverberating activity. Activity continues after an input has gone. And finally, encoding. This is the transient or stable plasticity that occurs between connections. And what's emerging from our data is that, remarkably, all three forms of the signal processing are different across the various hippocampal nodes. And in the interest of time today, I'm really going to focus on signal transformation and how this differs across the specific nodes of the hippocampus. So during this talk, I'm really going to try and make three points. The first one is, and provide examples of these, and try and convince you that there's novel neurobiology underlying these processes. So we have signal processing at the inputs of cells, signal processing at the output of cells, and then a signal amplification via local circuit arrangements. I'll then provide a sort of speculative hypothesis about how these different forms of signal processing enable multiple modes of operation in the context of episodic processing. So signal transformation can occur at one of two sites. At the synapse, the input to the neuron, you can have changes in the response, either increasing or decreasing. And again, then across the somatodendritic domain, how this response influences spiking. In this part, I'm just really going to focus on the presynaptic, the synaptic input. And classically, these factors are regulated by processes within the presynaptic terminal. And you see classic frequency facilitation of those synapses that have a low release probability, and a depression at those synapses with a high release probability. And all we wanted to do initially was see how does this processing differ across three different nodes within the hippocampus? And we focus on the CA3 to CA1 synapse, and the two direct inputs to the dentate gyrus, the medial and lateral perforant path. And what we found was that these are all very different. As I alluded to in the first slide, these processes differ across all the different nodes. So the CA1 shows the classic frequency facilitation. The medial perforant path shows a depression. This is in response to a 50 hertz or a gamma stimulation. And the lateral perforant path is the real oddball here. It shows an initial facilitation, which is followed by this very pronounced depression of the response. And while this phenomenon has been reported both in vivo and in vitro previously, the mechanism underlying it has not been investigated, but also how unusual it is has not really been discussed. It's very unusual to see this kind of response. So we are rather, a graduate student in the lab, Mr. Julian Quintanilla, has been investigating this in more detail. And what we find, or what he has found, is that the lateral perforant path is operating as a low pass filter. At lower frequency inputs, information flows straight through. However, as the input frequency increases, we see this filter kicking in within the train. If we modulate the release probability of the synapses by lowering the extracellular calcium, we can remove this filter. So things start to go through. So this gives us some kind of clue as to what may be going on. And in order to kind of explore this further, we turn to computational simulation. And this is some work by Dr. Connor Cox in the lab. And in this simulation, we model the various factors that are critical for driving release at a synapse. And using the experimental constraints, this model predicts that vesicle refilling, so this stage of the process, is a key variable in this response that we're seeing. So we then wanted to see, is there any unusual molecular changes in these terminals? And this is some work by Dr. Julie Lauterborn. And what this shows is that the two transporters, the two primary transporters for vesicle refilling with glutamate, vGlut1 and vGlut2, have very different rates of refilling, with vGlut2 being significantly slower. And what this data from Dr. Lauterborn shows is that the expression of vGlut2 within the terminals at the lateral perforant path are much, much higher, suggesting that once this readily releasable pool becomes depleted, these vesicles will be unable to be refilled quickly enough to meet the demands of high frequency activation. So this provides a novel mechanism for why this synapse is filtering. If we move now on to point 2, I'd like to talk about a specialized output filter within field CA3. So now we're moving down from the dentate gyrus down to the subsequent subfield, and we're recording the response to stimulation of the CA3 synapses, so within the associational system, and then the output of this structure in CA1. And again, if we bring up this schematic of signal transformation, and I should like to point out at this point that typically what we would expect to see, if we see a facilitation of the synaptic response, we would typically expect to see an increase in the output from the cells. They tend to follow very closely. So if we look at the presynaptic responses in these two different synapses, we see that both of these show frequency facilitation, although to a slightly different degree. This is doing what we would expect at 25 hertz, so at an input in the beta range, we see frequency facilitation. However, when we look at the output in the form of a population spike, the CA1 response is, as we would expect, we see a marked and pronounced facilitation of the response. However, within the CA3 associational system, so within these synapses, we see a really strong filter on the output. And this was really surprising to us, and it's not something that's really been reported within the literature prior to this. And it suggests that this CA3 output filter, again, is operating as a low pass filter and is engaged at frequencies above the beta range and above. So an obvious candidate for what may be responsible for this filtering is the engagement of the inhibitory system. And we wanted to explore this, the idea that that in some way within CA3, interneurons are becoming activated, and this is shunting the depolarization in the dendrites to prevent spiking. So we tested this with a GABA-A receptor antagonist, and what we found in the dendritic response was a modest increase in the frequency facilitation, but at the output level, the population spike was almost normal throughout the train. So this suggested to us that, yeah, inhibition is important in this filtering that we're seeing in field CA3. So an interesting question, as you remember, is this worked in CA1, right? This frequency facilitation was able to drive output. So what is different at the local circuit level between CA3 and CA1 that may underlie this phenomenon? Both of these fields have similar repertoires of inhibitory interneurons that innervate specific subcellular domains of the pyramidal cells, as you can see here, dendritic versus somatic. What is it that may be different? And we explored this briefly and found that there is a population of somatostatin interneurons located in CA3 that occupies a site in stratum radiatum that are absent in CA1. And so our initial hypothesis is that maybe these interneurons are critical for shutting down spiking within CA3, and not being in CA1, they're not functioning there. So this is an area we're exploring, and we can manipulate this down the line. So moving now to point 3, looking again in the same system within CA3, I'm going to talk now about amplification, again through this local circuit operation. And if we stimulate the CA3 associational system this time at 5 Hertz, so in theta frequency, we see the converse. We see no enhancement of the synaptic response, but we see a pronounced amplification of the output. So this is, as I said, quite an unusual observation. It's not probably what we would expect. And so how could this be doing it? And we postulated that maybe activating the network in the theta range actually triggers the local recurrent activity such that you're driving feed-forward excitation rather than inhibition. And as you can see here, we looked at the spiking that occurs within a stimulation train, and we saw on the first pulse very little spiking between pulses. But by the final pulse in the train, you can see a significant increase in spiking between pulses. And so this sort of led to the hypothesis that on the initial stimulation, we have little or no input from the commercial associational inputs within CA3. But following repetitive stimulation, this recurrent circuit becomes engaged, priming the cells on subsequent inputs to spike. Now, you probably have noticed during the talk that I've talked about stimulating the CA3 associational system directly. And in fact, that's not how it works in the real world. It receives a direct input from the entorhinal cortex, in this case by the lateral perforant path, and an indirect input via the mossy fibers, or a combination of both. So we next wanted to look at, can we activate the CA3 associational circuit through these physiological inputs? In order to do this, we used a hippocampal slice prep, where we were able to activate the direct input in isolation, the indirect input by the mossy fibers, and both together. And what we found was really quite remarkable. When we stimulate each input individually, so the lateral perforant path or the mossy fibers, we saw a modest amplification of the output within CA3. When both inputs were activated together, we saw this really robust amplification of the response within the train. But again, this is not the only thing. I should really point out that all of these recordings were made in the distal portion of field CA3. If we record simultaneously from the proximal portion in CA3C, we actually saw that this amplification was only evident in the distal portion. Okay, so it suggests that there's an amplification along the proximal distal axis of CA3, which was really quite startling for us. It also shows here that when we engage the network at a higher frequency, which we think is what we showed engages this very robust filter within the associational system, throughput is completely impaired, suggesting that everything is working when we hook it up as it would be. So to sort of summarize and speculate somewhat, I've shown you here 3 features of signal processing that differ across the hippocampal nodes. We've got this strong input filtering at the lateral perforant path to dentate gyrus synapse. We have within CA3 an unusual output filter and amplification by the same circuit that occurs in a frequency dependent manner, which is, I think, quite a neat little trick. And what we can take home from this really is that inputs to hippocampal circuits in the range of theta are really optimal for driving throughput, so for information to flow through the circuit. And this raises a suggestion that input frequency may be critical for determining the operational modes that this structure functions at, and these operational modes are likely to be critical for the demands of episodic processing. So in terms of throughput, this will mean rapid detection of novel versus familiar context and cues, right? But what does this mean for the higher frequencies, right? We know that the hippocampus and the entorhinal cortex operate at these higher frequencies or combinations of. So, oh, I forgot about this, the throughput. So this is an example, a further example of the throughput. If we move down into field CA1, again, stimulating both inputs, you can see this facilitation. So this output from CA1 is greatly amplified, so suggesting that it is really amplifying at theta. So back to the higher frequencies that I jumped to too early. What do we think the higher frequencies might be doing? And at the beginning of the talk, I mentioned cycling and plasticity as three processes that the hippocampus needs to do, presumably, for episodic processing. And our data that's ongoing suggests that it's these higher frequencies and patterns that are critical for engaging cycling, like behavior to the reverberation that I talked about earlier, as well as plasticity. And engaging these operations will be critical for episodic processing, for certain elements of episodic processing. And finally, I'd like to just bring up the notion of extrinsic factors. These are likely to be engaged by specific behavioral states, and these are likely critical for promoting both the cycling and plasticity elements of episodic processing, and add in actually a third signal transformation factor, which is something we are looking at now. So that would just lead me to thank everybody that's worked on this project. Those ones in bold have worked directly on data presented here, but other members of the lab are contributing to other elements that I haven't been able to discuss today. Thanks very much.